Erwin Chargaff in 1951 found that nitrogenous bases in DNA show specific ratios. They vary from species to species, but they remain same within the species. That is, DNA specimens which are isolated from one species have same base composition, but they vary when they were isolated from different species. And nitrogenous base composition in DNA doesn't change in given species with organism's age, nutritional state, or changing environment. So we can say that nitrogenous bases in DNA remain same throughout the life. It does not change with organism's age. And he also found that amount of adenine is equal to amount of thymine in DNA, and amount of guanine is equal to amount of cytosine in DNA. And this implies that total purines and total pyrimidines are in 1:1 in DNA. These are purines, adenine and guanine. These are double ring structures, two ring structures. These are purine. So number of purine is equal to number of pyrimidines. Thymine and cytosine are the pyrimidines. So this implies of 1:1 in DNA. And this is known as Chargaff's rule. And after this rule, Morris Wilkin and Rosalind Franklin, they were also doing X-ray diffraction analysis to see the DNA structure. And they were the first who claimed that DNA is a double helix molecule. Watson and Crick are the scientists who showed this DNA model in 1953. And they showed that DNA is a double helix. It is composed of two polynucleotide chains which are held together by the nitrogenous bases in it. These are the nitrogenous bases in it. Through these nitrogenous bases, these polynucleotide chains are attached to each other. These polynucleotide chains are basically anti-parallel to each other. This, for example, this one is 5' to 3', then the other should be 3' to 5'. So they are opposite in direction. You can see this one is 3' to 5', and the other one is 5' to 3'. So they both are opposite in direction. And in between these two polynucleotide chains, nitrogenous bases are present. For example, here is adenine bonds with thymine. Cytosine bonds with guanine. So adenine always pairs up with thymine by forming two hydrogen bonds, and cytosine always pairs up with guanine by forming 3 hydrogen bonds in it. These are 3 hydrogen bonds. So according to the number of hydrogen bonds, cytosine guanine bonding is more stronger than adenine thymine. So it requires more energy for its breakdown. Here you can see that the width of this duplex, this is the width of the duplex, it is 20 angstrom. And this is one turn of the duplex. Each turn in duplex has 10 nitrogenous base pairs, and each turn is 34 angstrom. Each turn's length is 34 angstrom. And this one is the major groove. You can see this one is a major and this one is a smaller one, so we call it minor groove, and the larger one is known as major groove. Both these polynucleotide chains are complementary to each other. Complementary, like adenine always pairs with thymine, cytosine always pairs up with guanine. So these are complementary to each other, and there is no restriction of the sequence of nucleotides along the length of DNA strand. Like after adenine thymine, it can be guanine cytosine or cytosine guanine, or maybe again adenine thymine. So there is no restriction of the sequence of nucleotides. and sequence can vary in countless ways. This sequence is specific for different species, organisms, and even individuals. Here you can see the model of DNA. These are the ribose sugars. These are the ribose sugars, right? Here they are present in this form, but in this polynucleotide chain, ribose sugars are basically altered. You can see now they are in their alternate form. So here you can see that at 5' end, phosphate group is present at carbon number 5, and at 3' end, hydroxyl group is present in this polynucleotide chain. But on the other hand, in this polynucleotide chain, at 3' end is upward and 5' end is lowered, and hydroxyl group is present at 3' end again, and the phosphate group is present at 5' end. Here, through these nitrogenous bases, these two polynucleotide chains are attached to each other. So you can see thymine is bonding with adenine by the 2 hydrogen bonds, and guanine is bonding with cytosine by 3 hydrogen bonds. As I told you that guanine and adenine both are purines, both are 2 ring structures, both are composed of 2 rings, and cytosine and thymine, these are pyrimidines and these are composed of 1 ring. Here you can see that how the DNA is presented. This backbone of DNA is composed of sugar and phosphate groups. Sugar and phosphate groups are hydrophilic, and their hydroxyl groups make hydrogen bond with the water molecules. And if we see inside, here are the hydrophobic nitrogenous bases are present. So this is how the structure of DNA is maintained, that phosphate group and ribose sugars are present at the backbone, so they are facing outside, and inside nitrogenous bases are linked to each other.